visit and meet all of you. And so I've been working with the Seattle Flu Alliance for the past couple of years on this project. It used to be called the Seattle Flu Study, so I might switch in between those two names. But I just wanted to briefly introduce in general what my research focuses on. And so I study the ecological, evolutionary, and behavioral drivers of respiratory virus outbreaks with aims to improve disease surveillance and also better understand and predict recurring and emerging outbreaks. And the general themes of research I've been working on the past couple of years, which the first one, which is what I'm talking about today, is looking at the role of human mobility in respiratory virus outbreaks, contrasting patterns observed in pre and post-pandemic years. I've also worked on linking the ecological and evolutionary dynamics of annual flu outbreaks in the US. And then lastly, I've been heavily involved in operational forecast and scenario production modeling through the Scenario Modeling Hub, and that's how I know the MOBS group, for example. But so just the motivation for this project that I'll be talking about today is that, as everyone's aware, COVID non-pharmaceutical interventions severely disrupted the transmission of SARS-CoV-2, but also endemic respiratory pathogens at a global scale. And so this figure demonstrates that both in the northern and southern hemisphere, despite continued testing for flu during the pandemic, which is this black line, there was barely any flu circulating during the first years of the pandemic. And then cellphone mobility model SARS-CoV-2 dynamics, but relationships between mobility and endemic respiratory pathogens is not as well understood. And understanding the role of mobility in endemic respiratory pathogen outbreaks is crucial for disease surveillance. And so disease surveillance and predictions. And so one example is that overlapping epidemics can put extreme strain on healthcare systems. And an example of that is the 2022-2023 triple-demic of COVID, flu, and RSV in the US. And then also it could be informative for the timing of interventions, not just lockdowns, but also vaccinations, immunoprophylaxis. And so the Seattle Flu Study was launched in the fall of 2018, and it's conducted intensive respiratory pathogen surveillance since then. And so this represents a really great opportunity to look at the impact of the pandemic on respiratory virus circulation, and also the potential role of human behavior in that. And so in general, I'll be talking about what are the impacts of behavior on respiratory pathogen transmission and which mobility indices may best capture transmission relevant contacts across pathogens and over time. And so I'll start off with the mobility data we use in this study. So we used a combination of data from SafeGraph and metadata for good. And the top figure is showing the percent change from baseline in behavior for different types of large-scale population movements. And so the top figure shows within neighborhood movement, between neighborhood movements of people coming into Seattle from other counties in Washington state, and then also people visiting from out of state. And we infer this based on people's home census block group and whether or not they're visiting points of interest within their own neighborhood or in census block groups like outside of where they live. And then the middle figure is showing the percent change in movement to visits to different categories of points of interest. And the last figure is showing the percentage of devices staying home in purple, and then the percentage of people masking in public. And that's based on survey data collected by the Delphi group at Carnegie Mellon in collaboration with Meta. And in all of these figures, there's a very thin blue vertical line, and that's a major snowstorm that happened in Seattle in February 2019. And then the vertical dashed line is Washington's state of emergency declaration. And then the stay-at-home period for King County is that light orange band. And what we noticed was that there was a drastic change in mobility before stay-at-home orders were even implemented. So like,
I'm very sorry, but I don't think your screen is sharing.
Oh.
So can you just
Oh,
confirm that you're sharing your screen or on Zoom?
Emily? I can try
Yeah.
again. Sorry about that.
Or I think maybe go onto Zoom and make sure that you're sharing.
Oh, okay. Sorry, rookie mistake.
That's all right.
Thanks for catching that. Here we go. That might be presenter view for people on Zoom. Can anyone confirm? Okay, I think that's should be good now, but I think that's
Yes.
okay. Okay.
You can even make it smaller.
Oh yeah, that's a good idea.
Picture, yeah.
There we go.
You can-
Stick it down.
Great.
Okay.
Buttons on the top.
And then okay there. Good.
Thank you.
And that's.
Okay, sorry, like several years into the pandemic, still figuring out sharing my screen. Okay, well, I'm sorry for the people on Zoom who couldn't see the visualizations, but okay. So now this is just showing the between neighborhood movement data, but just at a finer scale. So these are maps of Seattle at key epidemiological time points. Using the SafeGraph data, and the connections between the neighborhoods are movement of devices between census block groups. And the first figure is the February 2019 snowstorm. Then I show July 2019 just as an example of baseline movement. Then in the middle is the March 2020 stay-at-home orders. And then after that is the Delta wave in July 2021, and then the first Omicron wave in January 2022. And of course, predictably, like the stay-at-home orders caused like a profound, like dramatic reduction in movement between neighborhoods during the lockdown. But then what we observed for Seattle is that the mobility network didn't actually recover after the lifting of stay-at-home orders. You still see that these very strong connections between hubs in the network, but the network became much less dense. And so there was an overall decrease in the median number of connections between neighborhoods. And so that's what these bar or frequency plots are showing below. It's just the degree for each neighborhood. So here in this network, each neighborhood is a node. And this isn't weighted degree, this is just the presence, absence of connections. And so we found that the median degree declined after the stay-at-home period. And this particular study ends in summer of 2022. And the mobility network still hadn't recovered to the pre-pandemic levels by the time of the end of this study. But this isn't necessarily what's been observed for the US as a whole, like, or even Washington State. There's been, many studies looking at changes in the mobility networks over time. And so people have found that the mobility network is actually stable at a US level, but for just the city in particular, we found that was not the case. And just Seattle was a very COVID conscious city. And so now I'll give some background about the Seattle flu study. So it was a prototype pandemic surveillance platform It officially started in November 2018, and the whole goal of this study was to better detect and predict and study the spread of a pandemic pathogen in Seattle. And so it started about a year and a half. Sorry, I was getting confused with my my time, but so it started a year and a half before the pandemic. And also a big emphasis was taking testing beyond hospitals and clinics and into the community. So prior to the pandemic, there were kiosk stations in high foot traffic areas like tourist areas, transit stations, the Seattle airport. There were people could get tested in workplaces on the University of Washington campus. And then they started a pilot study of providing tests for people where they could request a test to be sent to their home, and then they would return the test and get a result for flu that had just started taking off at the time of COVID. So in January 2020, the study pivoted to study COVID. This turned into the swab and SIN study. So throughout the first 2 years or so of COVID, people could order a nasal swab from the study. It would be sent to their house. They swab themselves. They return it. It gets picked up by a delivery driver, returned to the lab, and they get the result for COVID within a very short amount of time. So the study actually filled like a big gap in testing at the very start of COVID, and the study did this in close collaboration with Public Health Seattle, King County. And so it was really important in detecting the spread of COVID at the very early stages of the pandemic. And for this particular project, I focused on the start of the Seattle flu study to summer of 2022. And there's about 81,000 specimens, respiratory specimens included in the study, but this is pared down from about 130,000. So I focused only on symptomatic individuals, and then also got rid of tests from multiple testing or people who didn't reside within the greater Seattle region. And so at the start, I just wanted to introduce to you that we saw 2 overall themes in our results, which were based on whether or not a particular virus has an envelope. And so the envelope viruses are flu, RSV, seasonal coronaviruses, and SARS-CoV-2. And the non-envelope viruses are rhinovirus, adenovirus, and enterovirus. And I forgot to mention that the study tested each swab for 26 pathogens. However, we only had enough positive detections to study about 17 of the endemic pathogens over time. But due to their lipid membrane, the envelope viruses are more susceptible to disinfection and hand washing, and they're less stable outside of the human body. And so we think that this contributed to some of the different patterns we observed during the pandemic in terms of the rebound of these pathogens after the stay-at-home orders. And so for estimating daily pathogen transmissibility, we reconstructed daily incidences from the PCR data from the study. However, we adjusted those data for daily test volume, the clinical setting, so hospital versus community, age group, so child versus adult, and then local syndromic respiratory illness rates. And then we estimated daily time varying effective reproduction numbers by date of infection, and RT is the number of secondary cases that arises from an index case, assuming conditions remain the same. And there's a lot of different approaches to estimating RT. In particular, we use like a semi-mechanistic Bayesian model, which I'm happy to talk about later more in depth if people have questions. But RT takes into account the cumulative number of susceptible individuals from the past, as well as the generation interval distribution. So that's the time from infection in the index case to infection in a secondary case. And we were able to map the observed cases from our surveillance study to infections by convolving over uncertain incubation period distributions and reporting delayed distributions. And one really cool thing about the study is it collected very detailed metadata for each person who was tested. So we have data on when someone first started feeling sick and when they were tested. And so we were able to estimate reporting delays down to even the pathogen level, depending on the number of positives we had for a particular pathogen. And it was also specific to the Seattle area. However, this is also conditioned on people seeking out a test or seeking out healthcare. And this figure on the right is showing the daily pathogen incidences are these larger curves in the back. The RT curves are the squiggly lines. And as I mentioned for the mobility data, you see this like drastic decline in respiratory virus circulation after the state of emergency declaration. And some pathogens took a long time to return to circulation. So the envelope viruses didn't really return until early to mid 2021, whereas these non-envelope viruses down here, on the far right side, they rebounded as soon as stay-at-home orders lifted. So now I'll talk about some pre-pandemic relationships between mobility and transmission. In the paper, we talk about some relationships we saw during this major snowstorm in February 2019, but I'll just talk about the season prior to the pandemic. And so I call these like the caterpillar plots, but it's the goal of this figure is to show a dynamic association between mobility and transmission over time. On the x-axis is week, and the points are the 5-month midpoint of moving window cross correlations between transmission and mobility. On the y-axis is the cross correlation coefficient, and then the points are colored by the lag between those two time series. And for this particular figure, I constrained it to just relationships where mobility is either leading or synchronous with transmission, because it can get kind of messy if you also allow for transmission to lead mobility. So the dark blue dots indicate mobility leads transmission up to 4 weeks, whereas the lighter colors are when you have more of a one week lead to synchronous relationship. And then we assess statistical significance of these cross correlations using time series block bootstrap permutations, where we shuffled each mobility time series for each window, and then to generate a null distribution of correlation coefficients. And we look to see whether or not the observed cross correlation coefficient was outside of that null distribution. So any of the filled in points means there was a statistically significant association. So we found for envelope viruses that large scale movements, such as between neighborhood movement, percent devices leaving the home, as well as child visits to child daycares, schools, and colleges either preceded or coincided with increases in RT, and the relationships were the strongest at the beginning of the season. And this makes sense because that's when there's the most susceptible individuals. This particular model doesn't account for susceptible depletion, but we assume that there's this weakening of the relationship due to susceptible depletion. And then I'm not showing relationships for the non-envelope viruses, but in general, we didn't really see any strong relationships between mobility and transmission. And this could be because non-envelope viruses circulate year-round, and they're less seasonal than the enveloped viruses in our study. So now I'll talk about relationships we saw during the pandemic, starting off with the stay-at-home orders and lifting of restrictions. And so this is showing SARS-CoV-2 cases as well as RT values over time, starting from February 2020 to I guess mid 2022, so the first few Omicron variants. And the orange line is the Oxford Stringency Index, which is a quantitative measure of stringency of MPIs that takes into account workplace closures, school closures, travel bans, gathering restrictions, but it doesn't measure compliance necessarily. It just takes into account whether or not measures were in place. And so this figure is showing those same like dynamic cross correlations, but just averaged by calendar month, and also allowing for positive and negative leads and lags between mobility and transmission. And what we saw is that in early 2020, there was a really strong relationship between mobility and transmission for the endemic pathogens. So for these first two, RSV and rhinovirus, but the patterns were very consistent across all 17 of those endemic respiratory viruses. So mobility and transmission really synced up at the time of the stay-at-home orders, which isn't surprising. However, for SARS-CoV-2, we found that mobility initially lagged and was inversely correlated with transmission. So this could be an artifact of, testing was just ramping up at the beginning of the pandemic, although we did put measures in place to make sure that affected our results as least as possible. But or it could mean that people were potentially adjusting their behavior in response to COVID restrictions or case counts. However, in April, May 2020, you do start to see this positive relationship between transmission and mobility for SARS-CoV-2. So now I'll talk about the individual COVID waves. And so during the lifting of stay-at-home orders, I mentioned that you do start to see this positive relationship between mobility and transmission. And then also during the winter 2020-2021 wave, we saw that visitor inflow, the percentage of devices leaving home, foot traffic to restaurants, people masking in public, and NPI stringency were correlated with SARS-CoV-2 RT. During the Alpha variant wave, we didn't see any significant relationships, but this was a pretty small COVID wave in Seattle. Then things sync up again during the Delta wave, and you see the same mobility metrics positively correlated with transmission. And then during the Omicron wave, mobility was not predictive of the increase in Omicron cases, but it did lead the decline of Omicron cases, suggesting a relationship between at least the decline in Omicron cases in behavior. However, we can't rule out that perhaps there was some sort of negative interference between the Omicron viruses and endemic respiratory viruses, or excuse me. Anyways, we think that just like the Omicron wave was just like so rapid, Omicron was so pervasive, just like there was no way, at least like with our mobility data, to catch any sort of like leading relationship. Now we'll talk about the rebound of the endemic viruses. So I mentioned that the non-enveloped viruses rebounded as soon as stay-at-home orders lifted. We think this could have something to do with their ability to resist disinfection and hand washing, and they persist on surfaces for longer periods of time. There's also other factors unrelated to the viral inflow, such as they tend to have longer periods of viral shedding. So there could have been people who were still infected with them during the lockdown period. And then as soon as contacts and movements increased slightly, they started onward transmission again. There's also a shorter duration of immunity. They circulate year round. Rhinovirus has a very high community prevalence already. And then also source sink dynamics. So some of those, those enveloped viruses, they may need to have external case introductions for outbreaks to be seeded again. Whereas these viruses, since they're very like pervasive in the community already, they didn't need those external case introductions to start recirculating. And in general, we found for the first 6 months or so a strong relationship between fluctuations in RT for these viruses and visits to religious services and restaurants, and then a negative correlation with NPI stringency. For the envelope viruses, I mentioned they didn't return until early to mid 2021, and we saw out of season outbreaks that peaked during the spring and summer months, and this is very consistent with what was observed worldwide for these viruses. And the rebound of these viruses coincided with just mobility metrics in general, so increases in within city mixing and visitor inflow into Seattle. There was reduced masking. Washington State also required students to return to school, or schools to at least provide a hybrid option in April 2021. And we see, I mean, it's just a correlation, but we saw that the RSV rebound occurred pretty soon after students started returning to school. And then one interesting thing is that although the relationships between mobility and transmission are, and this is very expected, were not as clear as what we saw for the stay-at-home period, they were actually stronger and longer lasting than what we observed during the COVID waves. And then I'm not showing this here, but during the Omicron wave, there was these drops in mobility that did actually catch the drop in endemic virus circulation during this Omicron wave. Okay, and so now I'll talk about sort of a supplementary analysis to the paper, but I thought people may be interested in this, is that so this study overall was focused on inference, looking at retrospectively looking at relationships between different types of behavior and transmission. However, we did try out some short-term forecasting of RT for SARS-CoV-2, as well as for 2 of the non-enveloped viruses that circulated continuously throughout the study. And so we used an approach adapted actually from a paper published by Mauricio back in 2015, where we have a moving window lasso regression model that selects which covariants are most relevant to transmission, and then projects 7 days forward. And we use 1 month moving training windows for this model because we found that those produce the best or most accurate predictions. However, with that short of a training window, there wasn't a clear pattern in terms of these particular mobility metrics are retained by the model in each window over time. But the different covariates we considered for SARS-CoV-2 were all the different mobility metrics, co-circulation of other viruses, so to take into account viral interference, climatic variables such as precipitation, temperature, the emergence of new variants, and vaccination, as well as past viral activity over the past 2 weeks, so 14 autoregressive terms. And then we compared different flavors of this model, incorporating different covariates to a baseline model, which was the autoregressive model. And we found that for SARS-CoV-2, a model including mobility actually had kind of a slight improvement, 13 to 15% improvement in performance, so lower root mean squared error during the stay-at-home orders, and also during 2021, 2022, which coincided with the emergence of new variants and vaccination. However, over the entire study period, mobility, including mobility, did not really improve predictions over just the baseline AR model. And so we concluded from this that mobility can provide a small to moderate benefit for forecasting. However, what it contributes pales in comparison to just knowledge of past disease incidents. And this is actually very consistent with another study published last year that used a different forecasting approach. They used transfer entropy to forecast COVID cases and deaths in Europe, but they found very similar results. So in conclusion, we found that mobility is most predictive during the stay-at-home period, so periods of dramatic behavioral change. However, smaller scale shifts in mobility also correlate with the onset of epidemics And then also with shorter interruptions to human behavior, which I didn't cover in the talk, but we found relationships during this pre-pandemic snowstorm and also during the Omicron wave. And the rebound of endemic viruses was very heterogeneous, but we found stronger, longer lasting relationships between mobility and transmission compared to SARS-CoV-2. And so the take-home message is like things are complicated. Mobility does seem to approximate transmission relevant contacts. And there's all sorts of caveats with the particular mobility data we use, which I'm happy to talk about. But due to time, like I saved that for the end in case people want to talk about it. But this is foot traffic data. It's not measuring spatial co-location of devices. And it's also for a metropolitan area. So the relationships we observed may not translate to rural locations. So it seems like mobility data is useful potentially. However, relationships between mobility and transmission vary depending on the pathogen, the magnitude of mobility change, and then the phase of the epidemic, where we see stronger relationships at the beginning of epidemics. Now I'm going to talk about like the very preliminary results for an extension of the study where we do actually take into account immune dynamics. So we consider the effects of both waning immunity and decreased social distancing on the rebound of RSV in Seattle for this extension. And so this figure is showing RSV seasons in Seattle using hospitalization data. And this dark gray shaded area is the average number of weekly hospitalizations from 2017 to 2019. And then this pre-pandemic season with the pandemic at the end is this dark line right here, 2019, 2020. And then RSV rebounded in summer of 2021. And however, it wasn't like a huge outbreak. And we think this could have been because there was still a lot of social distancing measures and masking in place. However, in 2022, there was a huge RSV outbreak that was much larger than past seasons, and it actually peaked 2 months earlier than what was typical for pre-pandemic seasons. And so the reason why we're further investigating RSV is that the epidemic dynamics are intrinsically nonlinear, and fluctuations in immunity and susceptibility could obscure relationships between extrinsic factors such as mobility and epidemic timing. However, if you use a dynamic mechanistic math model, you can explicitly account for the infection and immune dynamics, and also look at the potential role of mobility. So I just want to first mention that my colleague Chelsea Hansen at NIH received one of the CSTE grants to look at RSV vaccination scenarios for last season's respiratory, or the last respiratory virus season. And so she calibrated an RSV transmission model to King County hospitalizations and population dynamics, and then fit that model up until last season, and then did these different vaccination scenarios. And some background about RSV is that the immune dynamics are complicated. People consider the first few infections in life to be very important. Most people are infected by the time they're 2 years old. However, you can be reinfected multiple times throughout your life. It tends to cause like cold-like symptoms in adults, but it can cause severe respiratory illness in infants and older adults. So it is of high public health importance. And it's the most common cause of bronchiolitis and pneumonia in infants younger than 1 age. And last year, there's a continued interest, but last year, there's a huge interest in modeling vaccination scenarios for RSV due to the availability of new vaccines for pregnant women, for older adults, and then new extended half-life monoclonal antibodies for infants. And so that's what Chelsea's work was focused on, but she kindly let me adapt her model to look back at the rebound of RSV. And so this is a deterministic age-structured NSIRS model. So there's an extra compartment for maternal immunity. We modeled 13 age groups, and half of those correspond to infants younger than 1 year old. And this model was adapted from Pitzer et al. 2015, which I think is one of the seminal RSV transmission models. But there are some differences that have been incorporated compared to that 2015 model. And then the model assumes short-term protected maternal immunity, although there is a possibility of becoming infected during the maternal immunity stage. There's frequent reinfection throughout life, and then a gradual buildup of partial immunity following each infection. And so following each infection, individuals gain partial immunity that lowers your susceptibility to future infection. However, if you are infected again, you have a shorter duration and lower relative infectiousness compared to the first infection. And then also the risk of hospitalization is age dependent. I'm not going to go through like all this list of parameters, but this is just to show all of the parameters in the model. Actually, a lot of them are fixed because due to work from past studies. However, we do fit some of the seasonality and transmission parameters to be specific to Seattle. So the force of infection is influenced by seasonality, age-specific contact rates, as well as the total number of infected individuals and their relative infectiousness. And the calibration process it occurs in 2 steps. First, we fit the model to pre-pandemic seasons to estimate the seasonality parameters and reporting fraction. And then we refit it to the longer time series, including the pandemic period. And we scale the contact matrix by different mobility metrics, as well as a mobility scalar. And we refit the reporting fraction, assuming that reporting has increased during the pandemic period. And I also wanted to mention that for the contact matrix, we actually do use the polymod data, but I have considered other more recent data on age-specific contacts, but it actually doesn't really influence the results, the particular contact matrix. And for the mobility metrics, these top 4 are ones used in the previous study, and then the bottom 2 are 2 more network explicit metrics. The first one is called mobility network connectivity, and then the other one is median neighborhood degree centrality, and I'll explain those more on the next slide. So mobility network connectivity is the mean inverse shortest path length between neighborhoods in the mobility network for Seattle. And it's the inverse path length just because the algorithm assumes that a larger edge weight means a stronger connection, whereas that is actually the inverse for our particular use case. And this was inspired by a paper that looked at shortest path lengths of the mobility network of here is like a shortest path length tree for Berlin, but I think they, from what I recall, like the study was not just limited to Germany. And I also just wanted to put in a plug that I worked on this with some students at the Complexity 72 workshop that was held in Madrid this summer. And it's a workshop for early or young researchers in complex systems. And so you meet for 5 days and you form a team based on the students' interests, and then you work on a project really intensively for 3 days. And so I work with them on generating these more network explicit metrics, and then also incorporating the different mobility metrics into the RSV transmission model. And so what we found is that mobility network connectivity, and then also the inflow of visitors from other counties produce the best fits to the data. The contact rate here in orange is that rescaled contact matrix over time by mobility. And I just want to mention that this is very early stages. There's a lot of, I guess, like more work that needs to be done in terms of incorporating the mobility into the model, considering more complex relationships between mobility and contacts. There's issues with the mobility data itself in terms of the time frame it's available. So just want to emphasize this is early stages, but we did find these interesting results of connectivity between neighborhoods and then the inflow of visitors from other counties producing the best fits to the observed rebound of RSV, which is these last two waves. And so because these two metrics produce the best fits to the data, our preliminary conclusions or findings are that external case importations and the spatial spread of RSV in Seattle were most important, concerning influencing the timing of RSV rebound in Seattle. And I already mentioned next steps. We also need to do more to calibrate the model to observe the age distributions of hospitalizations. And one surprising thing is that the foot traffic to daycares and schools didn't produce good fits to the rebound of RSV. But as I mentioned, like we need to do much more with incorporating mobility data into the model, so that may change. And then I'm not sure how much time I have, but I just wanted to mention that I mentioned some of the other research I've been working on over the past few years. One paper that we had recently published, which was a years-long project in collaboration with WHO collaborating Centers for Influenza, was looking at the impact of antigenic drift on influenza epidemics in the US. So if anyone's interested in that and some like non-mobility or non-network related work, I'm happy to talk about that at some other time. And this was done in collaboration with the Bedford group at Fred Hutch. And then I forgot to mention at the beginning that Cecile Babu was my postdoc supervisor, and she's the head epidemiologist for the Seattle Flu Alliance, so I still work with her. So she's the senior author on this paper. And then I also mentioned that I've been involved with scenario modeling and also short-term forecasting of respiratory virus outbreaks. I first contributed forecasts to the Department of Defense, flu and COVID cases on military bases. Then I transitioned to forecasting flu hospitalizations. And so that's how I've gotten to know the moms group here. And in particular, I've been using like time series approaches for forecasting, which has been like an interesting exercise to apply that to trying to project flu hospitalizations for a whole season. But I'm also happy to talk about that work as well later if anyone's interested. So I'd like to acknowledge just like all the collaborators, like the Seattle Flu Alliance is a huge consortium of researchers, and so much work went into generating the data for that study. And then we have a paper on this mobility work that I spent most of the talk presenting on. And then I'd like to acknowledge Chelsea Hansen and Cecile Babu from the NIH for this more recent RSV modeling work, and as well as like my team of students from the complexity workshop for some of this early modeling work. So thank you.
Any questions?
So I have one question that is, I think a little bit overarching, because for me, it's not clear in the safe graph, unless I lost something, what level of data they could produce on kids and children. Because this is a problem then when you try to, when you say, you don't find correlation in the RSV.
Yes.
Is how you map those kind of movement in a sense. Yeah.
So this is my first limitation. So yes, that is a huge issue, is that, so the SafeGraph data, I think like legally, at least in the US, you cannot track the movements of cell phones of people younger than 16 years old. So like, how do you then use cell phone data to study transmission dynamics of viruses that tend to infect children. So that is a limitation for this first study, looking at like the dynamic relationships between transmission and mobility. We do see strong relationships at the beginning of these outbreaks between foot traffic to schools and transmission. However, I mentioned, for this preliminary RSV transmission model work, using a math modeling approach, we don't see a strong relationship. So yeah, I think that's like just an inherent limitation of this data. If anyone knows of any data for, I know that there's like more sort of, I guess, like cross-sectional data for contacts for children. But like in terms of like time series of how the contacts of children changed over time, I'm not really aware of the data source. So from this first study, we thought like, oh, it's promising that we do see at least like retrospectively a relationship between the start of the school year or foot traffic to schools, people starting to take their kids to daycare again, and the increase in these respiratory viruses that tend to infect kids. But yes, this is like an inherent limitation, is that the cell phone data don't actually track children. And so we're trying to approximate it with the movement of adults. And then, yeah.
And because this is also related to the difference between the urban and rural areas, because in rural areas, generally it's true that you have a lot of parents that bring the kids to school through carpooling or cars, etc. In the city, I would say that 12 years old, they are on their own on the T.
Yeah, in Seattle, it's a mix. Like there's a lot of private schools and public schools. I guess this data set would capture the older high schoolers, but you're right, like for kids who are like busing to school, their parents aren't going to be with them and their devices won't be tracked. So yes, I mean, do you all like have any data that you use to?
That's the right question.
Yeah. Do you have the data that you could share with me to like look at this in a better way?
The issue is that kids really don't have the mobile phone, so they are always underrepresented in a way that is, I don't know how, and this is my second question, in a sense, safe graph is another known probability survey, so in a sense it's not a panel, and that's why I think those kind of data were much better when you have to stay at home order, etc. You see those huge jumps, etc. When you are in different phases, the representativeness can be argued. And the point is did you try any way to generate a panel or to do something that is more representative somehow?
So I guess it may just be like semantics, but like I do adjustments over time to account at least for the size of the panel of devices over time. Although we can't, unlike the cubic data, for example, like we can't constrain it to like following the same set of people over time. There are like, for the time series of mobility, there's adjustments made into, the size of the panel of devices over time. I actually really tried to get spatial co-location data for this study to get hopefully a better approximation of like contacts that are actually relevant for respiratory virus disease transmission. I don't want to single out Matteo, but I've talked to Matteo in the past about like a cubic data set for Seattle. And then I actually like talked to the metadata for good people for like 2 months because a lot of people have used the metadata to look at to have a spatial co-location mobility metric during the pandemic. So when the privacy, I guess, laws changed and people had to more explicitly opt into sharing their locations, it profoundly changed their data collection. And so they currently provide data starting in like, actually conveniently, like right after this like study ends, the the Nature Communications paper study ends in June 2022, and they provide the data starting in like August. And I worked with them for like 2 months for them to try to figure out how to reconstruct a data set for the pandemic period. They suggested like, oh, you could contact other researchers at University of Washington who might have used it, because like you can't actually share the data across institutions. I contacted those people, they didn't have it. Meta couldn't reconstruct it for me. They just said like, they ended up saying like after months, like, we can still try, but like essentially they didn't save it. So I would really love to use spatial co-location data to try to get to better approximate like contacts, actual contacts that the foot traffic doesn't actually do. I mean, I shouldn't say it doesn't actually do it, because we do see, you know, at least like statistical correlations. We see some, you know, hints of relationships. I think it would be great to have spatial co-location data, but if anyone wants to help me with access, please let me know. But yeah, especially for this RSV rebound project that, you know, is ongoing. So.
Really impressive stuff. I have a lot of questions, which I can ask someone when say like, are there wastewater data that you can compare to? Because I'm curious whether that tells us anything. But I think what a lot of people are interested in is whether there's been interaction between SARS-CoV-2 and influenza. And it seems like you could look for that, not with SARS-CoV-2, but for like non-SARS human coronaviruses pre-pandemic. Your mobility forecasting results kind of suggest that there must be some interference going on or something else, because the mobility changes aren't explaining a lot of what's happening. But, and this is my actual question, looking at the caterpillar plots, it looks like there are different signals associated with mobility in the different settings, like between daycares and schools and some of the other places.
Yes.
And if you're forecasting things on the city level, like, could it just be sort of aggregating over lots of different signals with respect to mobility that somehow washes out the piece of mobility?
So I'll just first off, for like the supplementary forecasting analysis, we just did that for SARS-CoV-2, rhinovirus, and adenovirus, because they were circulating continuously throughout most of the study, and like the particular forecasting approach like requires like a time series of data to train the model on. And so there is a study that is still a preprint right now, where someone looked at from Seattle flu study, Roy Burstein, who's at IDM. He looked at individual levels, so like at the individual person, like data on co-infections and whether or not there's viral interactions. We're just now starting a collaboration with people in Lyon to look at more population level viral interactions, but it's not like a mobility explicit like study. One issue with this data set and like looking at viral interactions is that most of the people are symptomatic. And so I think for the study during COVID, where people could order a test and get tested and get their SARS-CoV-2 result back, 90% of the people in that study are symptomatic. There's like 10% asymptomatic. And so we're going to look in to see if whether or not we can incorporate those people. But I just bring that up in that there's been, for example, like I think Mark Lipsitch is the senior author of a recent paper about viral interactions and how there's all sorts of problems when you're trying to estimate these interactions using data just for symptomatic people. So and this is really a study that's like convenient sampling. It's not like demographically representative. We try to do like some reweighting of data to try to, counteract that. But so the answer is no. Like with viral interactions, I think it's like really difficult, at least for this study. Like for example, with like, say if like the UK REACT study had also collected data for other pathogens, I feel like that would be like the ideal study to look at viral interactions at the population level. But I have not personally like done any sort of integration beyond that, like little forecasting supplementary and analysis of like trying to take into account viral interactions. For SARS-CoV-2, at least, we didn't see like, it really was like, honestly, like having those autoregressive terms is what is driving the whole thing, and mobility provides like small to moderate improvements at certain time periods. The viral interaction component like never improved model prediction, but this is just, you know, modeling RT in one location. So for RSV, like flu, things like that, we haven't tried to like do any sort of like forecasting for Seattle. And so like, I agree with you. There's like, I think, well, what Alex brought up is like with these concerning like this data being able to track children, that could be a big issue with like the noisiness to the results. So yeah, sorry, that's like a sort of a rambling response, but yeah, I'm happy to talk more about it. Yeah, it's just like one aspect of this study is like, it's an amazing study. There's like so much data, but it is actually like convenient sampling. And so I think there's, in the past, I think there's a lot of criticism of studies like that, just, it's a convenient sample of symptomatic people. And there's a lot of criticism of just estimating viral interactions with symptomatic people. So I don't know if we can do that with this data set, but we're actually starting a new project to try to do that, but it's not mobility related, at least not yet.
Thank you for the presentation. My question is about your very first slide, the long-lasting changes in the mobility data. So it never bounced back to its previous. Yes, that's going to be that's fine. So did you have a chance to look into the mechanisms for this long-lasting change, specifically once you separate the part that is about, you know, still people being cautious back in 2022, not going out as much, and also removing the part that's about working from home, right? So, yeah, does it leave a significant portion to suggest that some structural changes in people's behavior in terms of mobility has happened, which might still be observed today?
I haven't looked at mobility, like the present day mobility for Seattle. So in terms of looking at other like behavioral factors that could be influencing, you know, like this like dramatic, like this decline in between neighborhood flows that doesn't really recover by summer 2022. I haven't looked at, I think it's sort of like the general maybe like hypothesis is it could have something to do with like COVID consciousness and a lot of people from in Seattle working from home. But no, I haven't quantitatively like done any sort of analysis looking at like what's actually sort of like underlying determinants of like the network, it's network structure, changes in the network structure itself. Like I haven't explored that.
An interesting question is to whether COVID has resulted in some kind of sustaining more segregation among people, even beyond working from home.
Yeah. No, I have not personally looked at that. I just have like colleagues who have, not in terms of like segregation, but like I've worked with a postdoc in Trevor Bedford's lab to look at like SARS-CoV-2 genomic epidemiology at the state level, where we incorporate mobility data and look at flows between counties. And it actually is like the state level network is stable. And it's actually produces pretty similar results to like commuting behavior. And then I think she's presenting here in a couple of weeks. Julia Polano has looked at the mobility network at the national level for the US and has found it's actually very stable throughout the pandemic. But no, it's a very interesting like I haven't personally looked at it, but no, it's a very interesting question. I just haven't like looked into it myself in terms of like sort of like, I mean, I did think this was like one of the more interesting like findings, even though it wasn't like the focus of the paper itself. And yeah, if anyone is interested in like working on this, or I just haven't like personally worked on it, but yeah, I think it's really interesting because also I don't know about more long-term studies of mobility in other cities that are as like specific like this, but there's been other papers that have looked at relationships between mobility and COVID across the US, like at the county level and things. And there are other cities where in the particular paper I'm thinking of is looking at the early stages of COVID. They actually found that like in San Francisco, for example, there was this similar like unexpected negative relationship between COVID cases, or I'm not sure what COVID metric they use, but COVID cases and behavior. And so I do think that there's something going on in Seattle, but like at least at the state or national level, it doesn't seem to have had a lasting effect for the US.
Okay, we have one question on Zoom from Esteban, so we're gonna,
Okay.
Esteban, if you wanna go off mute, we can see if we can hear you.
Hi, thank you. Thank you so much for the talk. Can you hear me, Amanda?
Yes, I think we can now.
Okay, thank you. Thank you for the talk, Amanda. Yes, so one comment, we have a paper about the impact of the mobility, the change of behavior on segregations after COVID.